Hello all of you. Today I would like to discuss about the term fluorescence, which is a special property of fluorescent molecule, and fluorescent molecule of our interest here is photosynthetic pigment. In this we will learn how the photosynthetic pigment by using fluorescence phenomenon transfer light energy from one pigment molecule to another. In the next part of my presentation we will talk about the mechanism of different reactions in photosynthesis. So let us first talk about fluorescence. The phenomenon of fluorescence has greatly helped in our understanding of the transfer of light energy from between the photosynthetic pigment by resonance. And here resonance means increasing amplitude of oscillation of electromagnetic wave, that is light. Suppose this is the molecule and it is at ground state or lower energy state. Ground state or lower energy state of a particular molecule also indicates towards its stable state. So in next step we are going to provide an energy to this molecule which is in their ground state, and the energy should be in the form of light energy. Here energy can be calculated through the formula E is equal to h nu, where h is Planck's constant and nu is the frequency of electromagnetic waves. So after absorption of light energy, the molecule which is in the ground state now reaches to its excited state. Excited state can also be referred to as higher energy state. So excited state and higher energy state of any molecule is also considered as unstable state. Now as you know that there is a tendency of every molecule that it wants to reach at its stable state. In this case unstable molecule also wants to attain its stable state. For that this molecule have to leave this excited energy level and it has to reach at its ground state. So now the molecule is coming back to its ground state and it is reaching to its ground state by releasing some energy in the form of radiation, right? And the wavelength of released radiation will be longer than the wavelength of absorbed radiation. As we know that energy is inversely proportional to the wavelength of radiation, which means that energy of emitted radiation is lower than the energy of absorbed radiation at ground state. So by keeping all these points in mind, now I would like to talk about fluorescence phenomenon in chlorophyll. So as I told you earlier that at normal state of a molecule or atom is called as ground state or singlet state. So when electron of chlorophyll molecule absorbs a quantum of light, it raised to its higher energy level, which is also called as excited second singlet state. This is unstable state, so electrons comes to next higher energy level by the loss of some of its extra energy in the form of heat, and this is called as excited first singlet state, and it is also unstable. From the first singlet state, the excited electrons may return to the ground state in two ways. First is either by losing its remaining extra energy in the form of heat, or by losing extra energy in the form of radiation energy. So the latter process is known as fluorescence. The substance which shows this property of fluorescence emit fluorescent light only during the period they expose to incident light. Secondly, the fluorescent light is of higher wavelength than the incident light. So fluorescent light has lower energy than incident light. Here one question comes that why fluorescent light has lower energy than incident light. The answer is some energy is used to lost during the change of excited second singlet state to excited first singlet state in the form of heat energy. So here I would like to tell you that fluorescence has greatly helped in our understanding of the transmission of light energy in between photosynthetic pigment. Photosynthetic pigments are very closely packed, and some of them absorb light energy at shorter wavelength with more energy, while others are capable of absorbing light rays of higher wavelength with less energy. So the pigments which absorb light rays of shorter wavelength will emit fluorescent light of higher wavelength with lower energy, and which in turn will be absorbed by other pigments which normally absorb light rays of higher wavelength with lower energy, right? So by this way, fluorescence helps in transmitting the light energy from one pigment molecule to another in chloroplast during photosynthesis. So this is all about the important role of fluorescence in photosynthesis. Now I would like to talk about the mechanism of photosynthesis. So mechanism of photosynthesis consists of two parts. First part is light reaction or photochemical reaction, or it can also be known as Hill's reaction. This part of photosynthesis is also known as preparatory phase because all the assimilatory power like NADPH and ATP are used to synthesize in this part of photosynthesis. Here the assimilatory power that is ATP and NADPH produced in light reaction help in the fixation of CO2 in dark reaction. And light reaction generally takes place in grana of chloroplast, and it takes place in presence of light. So light is most important for this part of reaction. In this reaction, ionization of H2, or you can say splitting of H2O in presence of light generally takes place, which results in the formation of proton and evolution of oxygen. So this process is also known as photolysis of water. Another part of mechanism of photosynthesis is dark reaction or Blackman's reaction or path of carbon in photosystem. Since with the help of assimilatory power produced in light, CO2 fixation takes place in this part of reactions to produce carbohydrate. So this part of photosynthetic mechanism is also known as payoff phase. This part takes place in stroma of chloroplast, and this part is independent of light, which means that it can occur either in presence or absence of light provided that assimilatory power are present. Now I would like to discuss about light reaction or preparatory phase of photosynthesis in detail. So the first important step of the light reaction is the absorption of light energy by chloroplast pigments. So here different chloroplast pigments absorb light in different region of the visible part of light spectrum. And second important step of this light reaction is the transfer of light energy from accessory pigment to chlorophyll A. Here, except chlorophyll A, all photosynthetic pigment are considered as accessory or antenna pigment. The light energy absorbed by them is transferred to chlorophyll A, which alone takes part in primary photochemical reaction. Here one thing you will have to take notice that chlorophyll A and B molecule can absorb light energy directly. In pigment system 2, the photoreaction center generally contain both chlorophyll A and B and absorb light at wavelength 680 nanometer, and so that photoreaction center are known as P680. And in pigment system 1, the photoreaction center generally contain chlorophyll A with few or no chlorophyll B, and it absorbs light energy at wavelength 700 nanometers. So the photoreaction center is known as P700, right? Now the third important step of this reaction is activation of chlorophyll A molecule by photons of light. Here P600 or P680 and P700 forms a chlorophyll A molecule in two pigment system, receives a photon of light and it becomes an excited molecules having more energy than ground state energy. During the fall of excited molecule to ground state, these chlorophyll A molecule expels an extra energy with electrons in both pigment systems. So this expelled electron takes part in electron transport chain in chloroplast. So regarding electron transport chain, explain electron transport chain I will tell you in the latter part of my presentation. So this electron transport chain generally helps in the formation of NADPH and ATP, which is also considered as the assimilatory power. Now I would like to talk about the fourth important step of light reaction. This step is photolysis of water. And in this step, splitting of water generally takes place in presence of light. So this step is associated with oxygen evolving complex or water splitting complex in pigment system 2. In this step, when pigment system 2 receives light, the water molecule splits into protons, electrons and oxygen molecule, right? And proton is generally utilized in the formation of NADPH. An electron helps in reducing the oxidized P680 in presence of light. Here oxygen has been released as waste product. Now the fifth step is electron transport and production of assimilatory power, that is NADPH and ATP. As I already told you, when chlorophyll A molecule present in photosystem 1 and 2 receives a photon of light, it becomes excited and expels an extra energy along with electron, right, in both pigment system. This electron, after traveling through a number of electron carriers, these are the electron carriers, is either cycled back or is consumed in reducing the NADP to NADPH. The extra energy carried by electrons is utilized in the formation of ATP molecules at certain places during its transport. This process of formation of ATP from ADP and inorganic phosphate in photosynthesis is called photophosphorylation. Now I would like to discuss about the type of electron transport and photophosphorylation in chloroplasts. So there are two types of these are of two types. First is non-cyclic electron transport and photophosphorylation, and second is cyclic electron transport and cyclic photophosphorylation. Now cyclic electron transport and photophosphorylation involves both photosystem 1 and photosystem 2, but cyclic photophosphorylation involves only photosystem 1. So let's discuss about non-cyclic electron transport or non-cyclic photophosphorylation, or it can also be known as Z scheme. This process is generally initiated by the absorption of photons of light by P700 form of chlorophyll A molecule in pigment system 1, right, which gets excited and electron is ejected from it, so that an electron deficiency or a hole is left in P700 molecule. This ejected electron is trapped by ferrodoxin, which is iron sulfur protein. From ferrodoxin, the electron is transferred to NADP plus. This is NADP plus, and as a result of the reduction of NADP plus, NADPH forms, right? Now let's talk about another type of now, sorry for interruption. Now when a photon of light is absorbed by P680 form of chlorophyll A molecule in pigment system 2, it gets excited and an electron is ejected from it, so that an electron deficiency or a hole is left behind in P680 molecule. The ejected electron is trapped by pheophytin here, and from pheophytin the electron passes downhill along a series of compound or intermediate electron carriers and ultimately received by pigment system 1, where it fills the hole. The series of compound which are the carrier of electrons are plastoquinone, cytochrome B6F, plastocyanin. At one place during the electron transport, that is between plastoquinone to cytochrome B6F complex, there is enough change in free energy which allows phosphorylation of one molecule of ADP to one molecule of ATP molecule, right? And this process is known as photophosphorylation. So if anybody will ask you where photophosphorylation takes place, then your answer should be in light reaction of photosynthesis. So hole or deficiency of electron in photosystem 2 is generally filled by the electron released during photolysis of water. And the proton which has been released after the splitting of water in presence of light is generally utilized in reducing the NADP plus to NADPH. So this point is also very important. Now I would like to discuss about cyclic electron transport or cyclic photophosphorylation. This process is also so as I told you earlier that in contrast to non-cyclic electron transport, the cyclic electron transport involves by photosystem 1, and it takes place if the activity of pigment system 2 is blocked. The pigment system 2 can be blocked by the use of a specific inhibitor or by using wavelength greater than 680 nanometers. So when pigment system 2 has been blocked, then these conditions arise, that is only pigment system 1 remain active. Second is photolysis of water does not take place. Third is blockage of non-cyclic ATP formation causes drop in CO2 assimilation in dark reaction. And fourth is there is a consequent shortage of oxidized NADP+. So in this process, when P700 molecule is excited in pigment system 1 by absorbing photon of light, the ejected electron is captured by ferrodoxin, and from ferrodoxin it ultimately falls back to P700 molecule, involving a course of number of intermediates like cytochrome complex and plastocyanin. At two places during the electron transport, that is between ferrodoxin to cytochrome B6 and from cytochrome B6 to cytochrome F, there is the phosphorylation of ADP molecule to form one ATP molecule. and these two ATP molecules are produced in the cycle. Here electron ejected from P700 molecule falls back to the same photosystem, so the process is known as cyclic electron transport chain. And the phosphorylation which has been happened here is known as cyclic photophosphorylation. And the asymmetry power here that is formed so the asymmetry power are formed in the form of two ATP molecule. So now I would like to discuss about the difference between cyclic electron transport and photophosphorylation and non-cyclic electron transport and photophosphorylation, right? So cyclic electron transport system is generally associated with pigment system 1, and non-cyclic electron transport system is associated with both pigment system 1 and 2. Second difference is the electron ejected from chlorophyll molecule is cycled back in cyclic electron transport chain, but in non-cyclic electron transport chain the electron expelled from chlorophyll molecule is not cycled back. Third difference is photolysis. In cyclic electron transport chain, photolysis of water and evolution of O2 do not takes place, but in non-cyclic electron transport and photophosphorylation, photolysis of water and evolution of O2 takes place. Fourth main difference is phosphorylation takes place at two places in cyclic electron transport chain. That is from ferredoxin to cytochrome B6 and cytochrome B6 to cytochrome F. But in non-cyclic electron transport chain, phosphorylation takes place only at one place, and that is between movement of electron from plastoquinone to cytochrome complex. And the last important point is NADP plus is not reduced in cyclic electron transport chain, but NADP plus is reduced to NADPH in non-cyclic electron transport chain. So this is all about the difference between cyclic and non-cyclic electron transport and photophosphorylation in the light reaction. This is all about today's class. If you feel that you are not understanding anything regarding this lecture, please call me or message me. Thanks and bye bye.